This conference will now be recorded.
Welcome everyone. Good evening, ladies and gentlemen. I, Navamita, extend a warm welcome to all the esteemed participants joining us today. We are delighted to have you all here for this webinar on understanding and tackling drug resistance in triple negative breast cancer, organized by Biosetup Life Science. Let me enlighten you all about Biosetup Life Sciences. Biosetup Life Sciences, founded by Mr. Ardhudip Das Sir, it is a hub for scientific research and learning, approved by ISO 9001-2015 certified organization, having a multidisciplinary team of faculty and research associates. We are continuously working for the career upgradation of the students. We have successfully organized many workshop, courses, career counseling, session, competitive preparation, CSIR, IIT JAM, CUTPG, GATP, IISC, and DBT. So, ladies and gentlemen, it is my distinct honor and privilege to introduce our esteemed keynote speaker, Mr. Nishan Shade, for today's webinar. Mr. Nishan is a biomedical engineer and he completed his BTech from Delhi Technological University. And he is also a cancer researcher at the Tata Memorial Center, Advanced Center for Treatment Research and Education in Cancer. In short, ACTRIC, from Khagar, Navi Mumbai. He is also an incoming biomedical PhD scholar at Johns Hopkins University in Baltimore, Maryland, USA. His research at TNBC Actric focuses on triple negative breast cancer and specific molecular signaling pathway of drug-tolerant persister cells. Having a diverse academic background, Mr. Nishant has made significant contribution in the fields of advanced mathematics and bioinformatics. In addition to this research, publication, software and tools development, he has been honored with the prestigious Erasmus Mundus Scholarship. This allowed him to study advanced mathematics at the University of Darbegen, Hungary. Before joining the TMC Actorate, he gained valuable experience working at King's College, London, England. He is also associated with University of Delhi South Campus and Jamia Millia Islamia, New Delhi. So, ladies and gentlemen, a little announcement of a little housekeeping. Before we start our meeting, please don't share your audio and video during the session. If you have any question, please write it down, save it for the chat box, and after the session, we'll take it in and the last of the session. So, without taking much time, I would like to invite Mr. Nishant Shahade. So, the floor is open to you. Thank you.
Thank you, Navnita, for such a wonderful introduction. And another thing, you don't need to see me, sir. Just tell me, Nishant. That's enough. Okay. So is my PPT visible now?
Yes, sir.
No need to say, sir. Nishant is good. Okay. So very good evening, everyone. So today I'm going to give a brief introduction about understanding and tackling drug resistance in TNBC, and I will hopefully try not to bore you all. So I will try to explain all the things, all the complexity in a very simple way. Okay. If you feel boring, you can just raise your hand. I'll try to make the things simple. All right now. So let's start from the introduction. Many of you know about the cancer. This is one of the biggest problem of India, particularly of India because of our population, and it is worldwide problem. But in India, it has a specific issue actually. So TNBC. So cancer have a different like lung cancer and different kind of cancer. So this is one of those triple negative breast cancer. So as the name suggests, why it is triple negative? Like something is negative three times. All right now. So the three times, one is estrogen, another is progesterone, and another is HER2 receptors. Like all these three things are lacking in case of TNBC. That's why we call it triple negative breast cancer. And on the below side, there is a short chart or diagram about the breast cancer subtype. So about 13%. I have taken this data from Healthline. So about 13% is of all the breast cancer is triple negative breast cancer. All right now. So account for 10 to 20%. This is the general idea of all the breast cancer, and this is more aggressive and fewer treatment options compared to other breast cancer subtypes. More aggressive. So where breast cancer is start, as the name suggests, it start in breast, but this is the structure of breast on the left side. This is normal breast cancer. These are the breast cells and all areola, nipple duct, lobule, and then chest, stroma. So these are the muscles, and another thing is ductal carcinoma in situ. So here is a visible lobule and duct on the second diagram in the middle. So this is the normal duct, and abnormal cells in duct are present in the last one. So these abnormal cells causes breast cancer. So lobule are the gland that make breast milk, and cancers that start here are called lobular cancer, and that cancer which start in duct. So duct are small canals like come out from the lobule and carry the milk to the nipple. This is the most common place for breast cancer to start. Next bit is, what is our concern? So the concern are three. Mostly there are many concerns, but most of these are high recurrence rate and poor prognosis, and urgent need for innovative therapeutic strategies, and significant challenge in oncology due to limited treatment options. So these are the three major concerns. So I have taken some data about the seer stage on the left side, and 5-year relative survival rate, localized, regional, distant, and all stages combined. So let me come to the next PPT and then I will explain these diagrams. These are the data. 5-year breast cancer specific survival rate, source from Healthline, and these are from the race white, black, non-Hispanic, American Indian, Alsaka native, Hispanic, and non-Hispanic. So let's explain the TNBC first and then I will come to the diagram. So what is TNBC? As I informed earlier, the three are negative estrogen, progesterone receptor, and also do not make any or too much of the protein called HER2. Another thing, these cancers tend to be more common in women younger than age 40 who are black or who have a BRCA1 mutation. That's why in the last PPT, there was a here black, non-Hispanican, American Indian, Hispanican, so black. So death rate is high among black race, 27.1, and in the seer stage, localized 5-year relative survival rate is 91%. And the survival rate, 5-year survival rate for US women is increasing day by day because of our medication and researches and all. So characteristics are what? Higher grade tumors frequent in younger women, particularly those of African descent. And we are Asian, so chances are high for us to be affected from TNBC. And symptoms are what? Similar to other breast cancer, but more aggressive growth. So aggressive is the highlighting point among this. So what are the mutations that are responsible for TNBC? One is BRCA1 mutation. So TNBC frequently exhibit mutations in BRCA1 gene, which is involved in DNA repair mechanism. So this mutation can increase the risk of TNBC development and may also affect response to treatment. So BRCA1 gene involved in DNA repair mechanism, and if there is certain problem in this gene, then it will affect. Another is TP53 mutations. Alterations in TP53 genes are common in TNBC and are associated with poor prognosis and aggressive tumor behavior. TP53 mutations disrupt cell cycle regulations and promote tumor growth and metastasis. And another mutation is P10 loss of function mutation or deletion in the P10 gene. A negative regulator of PI3K pathway are implicated in TNBC pathogenesis, and P10 loss lead to hyperactivation of PI3K signaling, promoting cell growth, survival, and metastasis. Another is RB1 mutations. Mutation in the RB1 gene, a tumor suppressor involved in cell cycle regulation, have been identified in TNBC, and the RB1 alterations disrupt normal cell cycle control mechanism, contributing to uncontrolled proliferation and tumor progression. So till now, we have discussed 4 mutations. One is BRCA1 mutation, another is TP53 mutation, another P10 loss, another RB1 mutation. Let's come to another EGFR amplification. So EGFR means what? Epidermal growth factor receptor, EGFR. So EGFR amplification is observed in a subset of TNBC cases and is associated with more aggressive disease and poor prognosis. So EGFR signaling promotes cell proliferation, survival and invasion, driving tumor growth and metastasis. So what does it? EGFR signaling promotes cell proliferation, proliferate hoga, and then survival, and then invasion, and then driving tumor growth and metastasis. Another one is PIK3CA mutation. Like maybe someone who is new and listening all these mutations, as the name are very odd. These mutations are also very odd, and we are still figuring out to solve these mutations. So mutation in the PIK3CA gene, which encode a key component of PI3K, AKT or mTOR signaling pathway. Actually, mTOR is one of my favorite topic, and I am looking forward to doing in this part. Are found in a subset of TNBC cases. mTOR signaling pathways are very interesting. If you are willing to understand this, then you are very welcome, and you can. These mutations contribute to aberrant cell proliferation and survival pathways, potentially driving tumorigenesis and resistance to therapy. So these were the mutations in the last 3 slides. These are the molecular pathway. Like for example, I am going to my institute from auto. So the auto will go to the road. So that road is kind of a molecular pathway. Institute will be another thing, and my home will be another thing. Home is A, institute is B, road is a line. Okay, scalar quantity, not vector. So molecular pathway. So BRCA1 2 mutations and homologous recombination deficiency. This is the molecular pathway. So BRCA1 role. BRCA1 and BRCA2 are crucial for the repair of DNA double strand break via homologous recombination. So mutation in these genes lead to HRD, homologous recombination deficiency, because there is a problem in homologous recombination. Double strand is getting break. So which contribute to genomic instability and tumorigenesis in TNBC. The prevalence in TNBC is what approximately 10 to 20% of TNBC cases harbor germline mutations in BRCA1 or 2 genes. And therapeutic implications.
Excuse
Patients
me,
with
sir.
BRCA, yes,
Sorry to interrupt you, sir. Sir, could you please explain more about germline mutation, sir?
yes, sure.
After the session, end of the session, we will explain each and every topic. Please write down into the chat box. For now, he
So
is
let
focusing
me,
on the.
yes, all right, Namita. So this is another molecular pathway. So therapeutic implication in therapeutic. So patients with the BRCA1 or 2 mutations are more likely to respond to PARP inhibitors. So these are inhibitors, for example, olaparib and talazoparib, and platinum-based chemotherapy due to their HRD status. So another pathway is PI3K AKT mTOR pathway. As I stated earlier, mTOR pathway, one of my favorite pathway. So even in actor at Tata Memorial Center, there are some scientists who are working on particularly on mTOR pathway, and even at John Hopkins, there are one or two scientists who are totally focusing on mTOR pathway. So pathway activation is what key aberrations in the PI3K or AKT or mTOR signaling pathway are common in TNBC and are associated with tumor growth, survival, and resistance to therapy. And this is therapeutic targeting PI3K inhibitors, for example, alpelisib and AKT inhibitors, ipatasertib and mTOR inhibitors, Overlumis are being explored in clinical trials for TNBC treatment. And other pathways are also, actually this is too much, and earlier I stated I don't want to make the presentation boring, so I will not waste your weekend. So another is immune checkpoint pathway, WNT beta catenin pathway, androgen receptor signaling pathway, Notch signaling pathway, EGFR pathway, and other emerging pathways. And if you are interested to understand about these pathways, then you are very welcome. I'll explain you even in this webinar, or personally you can contact me through LinkedIn or my WhatsApp number. I'll share in the last. So biological behavior of TNBC cells. What are the biological behavior of TNBC cell? So molecular and genetic heterogeneity. There are two things: homogeneity and heterogeneity. Hetero means different. Molecular and genetic heterogeneity. So genomic instability, like there is instability in genome. TNBC exhibits significant genomic instability, including high mutation rate, copy number variation. Key mutations often occur in TP53, PRCA12, and PIK3CA genes. And there is subtype classification. TNBC can be classified into distinct molecular subtypes such as basal-like, mesenchymal, and immune-enriched. These subtypes have different prognosis and response to therapy. So another is metastatic potential. Like in higher secondary, we have studied about metastatic, like which is moving to another place. and also epithelial mesenchymal transition (EMT), epithelial tissue, mesenchymal tissue, and all. So EMT, what is EMT? Epithelial mesenchymal transition. So TNBC cells often undergo EMT. So जो TNBC सेल है वो जाएगा EMT में, epithelial mesenchymal transition में. A process where epithelial cells acquire mesenchymal property, all right, and enhancing their migratory and invasive capability. And what will happen if migratory properties are enhancing? Then tumor will increase. The rate of increasing of tumor spreading will increase, in short. And circulating tumor cells, CTC, these are circulating. So high level of CTC are frequently observed in TNBC patients, correlating with metastatic spread and poor prognosis. And what is resistance mechanism? Chemotherapy resistance. There are so many of now way like chemotherapy and proton therapy. TNBC is often resistant to conventional chemotherapy due to presence of cancer stem cells and the activation of DNA repair pathway. So because the presence of cancer stem cells are there, and so it will affect the activation of DNA repair pathway. And if you are interested in cancer stem cell work, then you can go Dr. Rohan Khadilkar work at Actric Data Memorial Center. So they have done a lot of work there on cancer stem cells. And targeted therapy challenges. The lack of targetable receptors make TNBC less responsive to hormone receptor therapy and HER2 targeted treatment. And research is ongoing to identify new molecular targets and biomarkers. And emerging therapeutic approaches. Immunotherapy and the earlier chemotherapy. Immunotherapy, immune checkpoint inhibitor such as pembrolizumab and atezolizumab have shown promise in treating TNBC, particularly in patients with high PD-L1 expression. And PARP inhibitors for patients with BRCA1 or 2 mutations. PARP inhibitors likely olaparib have demonstrated efficacy by exploiting the synthetic lethality component. And combination therapies are also so combining combination where we are combining different things like combining immunotherapy with chemotherapy targeted agents or radiation is being actively explored to enhance treatment response and overcome resistance. And what we are discovering biomarkers. So ongoing research identify reliable biomarker for early detection, prognosis and treatment response prediction in TNBC. And personalized medicine are also, and a lot of researches are going on personalized medicine. So advance in genomic and transcriptomic profiling are paving the way for personalized treatment strategies tailored to the unique molecular characteristic of each TNBC patient. And let me tell you one thing, this is not included in the PPT, like biomarker discovery. As I hail from different academic background, from mathematical background and bioinfo background, and I've done a lot of work on advanced mathematics. So right now I am working on this is I'm with collaboration with some scientists. One is at IISc Bangalore and another is at from University of Illinois Chicago. So what I am doing, there are very few researches that is still going on related to what my idea is what. So I am joining two different domains. I can't explain the whole thing before publication. Let me tell. So what I am planning to incorporate quantum physics principles and biological phenomena. Like I am detecting biomarkers using quantum physics principles like dot structure and all. So this is kind of work that I am working. And another is personalized medicine. So I would like to acknowledge, and the last not the least, I would like to acknowledge Dr. Nandini Verma, my PI at Tata Memorial Center FTech, Dr. Mohan Chandra Joshi, under which I am working at Jamia Millia Islamia, and Dr. Christian Vasco, King College London, for guidance throughout the journey. and thanks to my mother and sister to always support me, and thank you everyone to be here for listening me. I hope I tried my best to not get you bored, and this is my email ID, so you can take a picture of it, so you can reach out to me, and we can do a collaboration. I'm very open for collaboration. You just give me, you just share your ideas. I will very happy to work with you, whether you are from this domain or from different background, whether you are from social science, from physics, bio. I don't. It doesn't matter to me. We can collab. We can get some ideas what we can do. That's my WhatsApp number. On the right side, that's my LinkedIn ID, so we can also contact here. So, and a special thanks to my younger brothers. So I can't let them away from my presentations. I always treat them as my younger brother. I'm an animal lover, so these all are my motivation to always be positive. One is Simba, another is Shiro, and Jack from DTU, and one of my new friend Chuchu. I adopted someone through from their house, so I just bring them. I just bring him at my home. So when it will get matured, I will release him, or he can live at my home as well. Thank you so much, everyone.
Thank you so much, sir. The session is open for question answer taking. So I would request all the participants, please drop your question into the chat box. So one by one, we could discuss the question and your queries also.
So Yashi has asked me, sir, can you explain the methodology steps you follow in this study in a nutshell? So in what nutshell you are asking me? Please explain it. Yashi, please explain. Okay. Yes, it's quite easy and very simple. Like what we do. Okay. Bioinformatics point of view. Listen, Yashi. For bioinformatics, first of all, we need data. So there are many public repository from where you can get data. There are cancer actors and all. So you can get data from there. But there are some restrictions in public data. So if it is publicly available, then you can use those. All right, and then what we will do is just grabbing all the data. Just water. So what we will do, data collections survey and public repository, then we will sample collect random sampling and purpose sampling. Okay, and then what we will do is statistical analysis and thematic analysis. So all these are the point of view according to bioinfo. Uh, Another question from Sonali Singh is, sir, can you explain HER2 receptor, also about BRCA2 and BRCA1 in DNA repair mechanism, also about BRCA2 mechanism? Wow, Sonali Singh, many question in a single. Okay. Dr. Dinu, I am again sharing the screen. Dr. Dinu now. Yes. So, Sonali, hmm, thank you everyone for such questions. Listen, Devrati Das, I am coming on Sonali Singh question. Sir, how is TNBC different from other type of cancers? In the first PPT, like TNBC, triple negative breast cancer, as the name suggests, there are three negative things: estrogen, progesterone, and HER2 protein receptor. So these are not present or are lacking in breast cancer. So that's why it's called TNBC. And there are many mechanisms behind this, but in short, we can define this by this. And yes, sir, can you explain HER2 receptor? So HER2 is what human epidermal growth factor receptor. It's a protein actually. So it will take part in epidermal growth factor receptor. So इसका काम क्या है ना कि ERBB2 जीन जो ओवर एक्सप्रेस हो जाता है in some cancer, particularly breast cancer. So when HER2 overexpress, it lead to increased cell division. So the cell division will increase. So in that case, HER2 positive breast cancer are more aggressive and can be treated with targeted therapies such as uh, Herceptin and all, and BRCA1 and BRCA2 uh, in DNA repair. So breast cancer type 1, breast cancer type 2 susceptibility protein are these. So genes produce protein that are responsible for repairing damaged DNA. So they play a significant role in maintaining the stability and for cells genetic information. And if there is some problem in cells genetic information stability, so it will goes like this. And BRCA2 receptor mechanism is also like BRCA1. So what we do in BRCA2 mechanism like recognition of DNA damage and recruitment of RAD51, the homologous pairing and strand exchange. So I guess Sonali Singh, your question is now solved out. So Harleen Kaur, who from like Harleen Kaur, I want to ask you that there may be a lot of receptor in the breast, but why they just focus on ER, PR, and HER2? All right. So, Harleen Kaur, listen, and very good. You are at AIMS Batinda. I've gone that campus. It's a good campus. So, reason is like there is significant growth in these threes, ER, PR, and HER2. One thing is like this, and there are implications for targeted therapy is also a reason. So estrogen receptors have a significant role in cancer. About 70 to 80% of breast cancer are ER. Uh, about 70 to 80% of breast cancer are ER positive. That's one of the reason, Arlene. And another role for PR is PR often correlate with ER. ठीक है if PR and ER are correlated, like if ER is present 70 to 80% in breast cancer cases, ER is there, so PR will correlate with ER. Another thing is like A, and ER and PR both positive tend to have a better prognosis and are more likely to respond to hormone treatment. Another is HER2 receptors. Like HER2 is a protein that promotes cell growth. So HER2 is around 15 to 20% of breast cancer. The HER2 gene is amplified, so leading to overexpression of HER2 protein, so which drive aggressive cell growth. So why they are important, like prognostic and predictive view, value, targeted therapy, and commonly in research. And about 70 to 80% is one of them. All right, Shalini. So Shalini, hello sir. I am okay. I am. So what you can do in colorectal cancer is like if you are good at machine learning and AI tools, so you can work on screening and diagnosis. You can make some tools and all which can help for better, like CT colonography and one is thing is I'm not sure you can confirm it. FOBT, fecal occult blood test. It is something. So it's a non-invasive test actually. So to detect blood in the stool, which can be early sign of CRC. So and another thing, actually, as you are a trainee here, so it will actually screening and diagnosis will take years. So you can work on biomarkers and molecular testing like on mutations detection. So it will be easy. So yes, you can work on that biomarker detection at your data. So Devarti Das. Devarti Das. Sorry if I mispronounce your name. I'm really sorry. Sir, on what basis are different cancers are caused? Like radiations are responsible for cancer, but how does it cause cancer in different part of the body? Actually, this I can't answer this question very particularly. There are many reasons responsible for this. Like not only even the naive person knows only why ki there are radiations that why cancer is causing, or someone is chewing tobacco, that's why it is causing cancer. But I have seen a lot of patients who never consumed tobacco and all the carcinogenic product we can say, and who and if someone who belongs from a rural area, so the chances are very low, like they affected through some radiations. So that is not the only reason. There are genetic reason also, like DNA damage, mutation, chromosomal aberrations, and reactive oxygen species ROS is also there. Like protein में damage हो रहा है. So that's why also not only the radiations are not only responsible, but there are certain studies which predict like someone is going from too many CT scans or MRI, so the chances are high because they are very harmful. And if you are in metro city where the scanner is present in the entrance where you are entering to the station, like there is a X-ray machine, I guess, X-ray UV, I'm not confirmed. So there is mentioned don't put your hand and all in this machine. These are carcinogenic. So just stay away from those. Sir, can you please give me some idea for BTech Biotech student how to get a job or internship or required skills? Listen, Sonal, if you are applying for a job, so it depends on you on what job you are trying to. If you are trying to get a job in this field, in biotech field, so you can go from some dairy areas and all. So for internship, and if you are particularly about any topic, like if you are interested in cancer, there are certain cancer institute you can apply there. So but never apply like a kid. If you are applying somewhere, so just try to be formal, like prepare your domain of interest in which you are interested, in which you are really interested, and you are well prepared. Like if someone asks you about something in which you have expressed your interest, like sir, I'm interested in this part and all, and I wanted to work under you, so they are definitely supposed to you, like they know these things in a better way. I know you are from bachelors, and it is obvious, like you know, not you don't know wide areas. It is obvious from a BTech student, from a undergraduate student. But if you are applying for something and you have expressed your domain of interest, then try to explain them, sir, I am interested in this, I have done work like this, and I wanted to work like this, I have such plan. So always be kind of like, sir, I have plan. I would definitely suggest you watch 3 Idiot movie. The Rancho have many ideas. So don't try to be like that topper guy, I forgot his name. So like think like Rancho. Okay, and if and another thing, Debarti Das, you have any, if you are wanted to better option, you can connect me on LinkedIn or WhatsApp. I will suggest you. So, because there are many questions. Uh, Dikshita Kulkarni, I had missed your introduction of the session due to weak network. Can you please go through your career pathway again? Sir, can you explain once again RBI mutation and give some brief overview of on heterogeneity? Thank you, sir. No problem, Sonal. And Dikshita Kulkarni, actually, I am currently I am currently working on a project at Tata Memorial Center Active, and currently I am working on three total projects. One project is at Tata Memorial Center Active, Navi Mumbai, and I am I have collaboration work with two projects. One is from IISc Bangalore. So at IISc Bangalore, I have collaboration with one scientist, Dr. Nikhil, and another is from University of Illinois Chicago. And next year I will join biomedical PhD at Spangler Lab. Okay, at John Hopkins University, Baltimore, MD. And I hail from a diverse background. So I have done work on advanced mathematics and bioinformatics. I was Erasmus plus student at University of Debrecen, Hungary, Europe for 1 year. So I got funded by European Union of around 10.5 lakh for 1 year, totally funded by European Union. So I have done studies on advanced mathematics there. So this is a short career pathway. So actually, most of the students, I am right now in cancer, and earlier I was in bioinfo and advanced mathematics. This is my mentality because I wanted to connect all the domains in a single thing so we can explore diverse background and we can solve the complex problem. Right now I am working on quantum physics and cancer. So this is something. Richita Mishra. Sir, how do we design cancer studies on diagnostic approach through bioinformatics tools? So, Narmada, Nikita, and Dr. Denu, I am coming on your questions as well. So, listen, design various question. Rachita, sir, how do we design cancer studies on diagnostic approach through bioinformatics tool? First of all, you define your objective and hypothesis and do a literature review in short, and go through several research papers. Okay, and don't run behind the from top publications. Like you are only considering nature. There are many publications who are not published in a good journal, who have a good reputation, but the paper is good. So you can take idea from those paper who are in not much uh, I don't want to go in that direction. So you can take idea from there and then collect your data from public repository and then bioinformatics analysis, genomic and transcriptomic data. And even now, most of the students are working on multiomics. What is multiomics? Genomic, proteomics, transcriptomics. So these are kind of, and then bioinformatics tools. You use those, and then what you can do is gene expression pathway and pathway analysis, machine learning, validation karo, internal, external, functional validation, descriptive statics, and all like this. So, can we run pedigree of triple negative? Is there any way track down more specific way to get a genetic marker? So, Dr. Dino, can we run pedigree of triple negative? Is there any way? Yes, it is possible actually. So to run a pedigree of TNBC to identify potential hereditary. So you what you need, first of all, you need to collect family history and then do genetic testing. So like BRCA1 and BRCA2 gene. So you need to understand the analysis of pedigree, and then what identify genetic markers. So whole exome sequencing and the whole genome sequencing and genome wide association studies, targeted gene panels and functional studies. So then you can do all these things. And what you should track down is key recruit a large cohort. Okay, so large cohort means power of genetic studies will increase if family is a large cohort. So the chances of getting accuracy and precision will be high. So you can and use machine learning and AI. And there are many softwares of ML and AI. Mm, in the earlier time, we used to write the whole code, but there are many ML and AI based tools which are helping to write code. So if you are very new to bioinformatics and programming, you can take help from those. It will guide you in a very better way. So myself, Ananya, IIT JM aspirant, but wanted to work in biomedical research field. So pathway, please. Listen, Ananya, you're IIT JM aspirant, so you are considering to do research field in India. So I'm not sure because I didn't try to get any research in India from my bachelor's. So I can suggest you from abroad. So if you wanted to understand, then please type. So so Rachita Mishra, what kind of methodology you wanted to know? Don't call me sir, please. Just tell me my name. I'm not too much older. Narmada S. Taloli. Why, despite ongoing research into nanoparticles and AMP, antimicrobial peptide, are these advancements not yet integrated into current therapy? Could leveraging these technologies potentially overcome drug resistance in cancer treatment? Listen, Narmada, there are many nanoparticles studies going on, but earlier nanoparticle studies were also causing carcinogenic. All right, you go to some papers like at Jamia Millia Islamia under Dr. Mohan Joshi. What they are working on nanoparticle is synthesizing a gold nanoparticle, because still according to right now studies, gold is not causing carcinogenic. So yes, we are doing work on nanoparticles, on gold nanoparticle, and it can be a good thing to overcome drug resistance in cancer. Another thing is what antimicrobial peptide are these advancements not yet integrated. These are working, but result are not still too good as per the expectations. Okay, Nikita Javale. Sir, I have completed MSc Geology Genetics. How can we apply the internship project? Just write your research proposal, make a good CV, impressive CV. Don't hosh posh on your CV. It should look good, attractive, and all. And your research paper should look formally. Always consider you are writing a letter to a scientist who have done a lot of work, so never try to make any mistake, any single mistake, so they can disappoint from you. So make a good research proposal. Try to approach them through LinkedIn, through mail, anyway. Go to many conferences. You will get a good contacts in conferences and all. So just go there. And if you are not able to go to international conference, go to their local conferences which are going to your place. So it will help you and all. And Nikita, you can connect me on LinkedIn. I will suggest you some good ideas. So Lopa Mudra Barik. So there are many protein receptors. Yes. So if we want to inhibit one protein receptor to treat a cancer, and how do we choose protein receptors to treat cancer? So listen, Lopa Mudra Barik. So actually choosing a protein receptor, if you want to inhibit protein receptor beta cancer, choosing listen, first of all you need to identify overexpressed or mutated receptor. All right. So IHC histochemistry. So you need to identify overexpressed or mutated receptor, first thing. And another thing is biological function and role in cancer को identify करना, assess करना. Another thing is evaluate drugability and existing therapeutics. क्लिनिकल रेलिवेंस देखो, सेफ्टी एंड साइड इफेक्ट देखना पड़ेगा, पेशेंट स्ट्रेटिफिकेशन एंड बायोमार्कर ऑल्सो, ऑल these things. And let me revise it again. Identify overexpressed or mutated receptors. Lopamudra. You need to assess biological function role in cancer. Pathway analysis and functional study. Pathway analysis is very common. Like if you are cooking anything, you need salt. So pathway analysis is like that salt. You need always if you are studying something. So do pathway analysis, understand its role in cell proliferation, survival, migration, and other cancer related issue process. Do pathway analysis and then validate the target. You should be focused on target validation. Lopamudra. You wanted to go home, you should remind the way from where you go to your home. So this is like that. Narmada S. Taloli, sir, how we can get into postdoc after PhD? They are you. There are many advertisement. Even PIs welcome a lot of time. Like I am open to take a postdoc in my lab. So getting a postdoc after PhD is very easy. They are welcoming you. You are not struggling. So many advertisement are there. So you can go to their websites. LinkedIn is a better option. Okay, Narmada, you got it now. So sorry, Ananya. I went to myself, Ananya, IT Jam aspirant, biomedical research field. So pathway, please type. So Ananya, I can suggest you from abroad. I'm not that person who can suggest you in India. So to be very honest, I will not guide you wrong. So even at John Hopkins, you just go to our website. There are the requirements. So even in US, you need to crack GRE, but GRE is not mandatory. So there are many labs who are admitting student without GRE score, but I have GRE score as well, and I had IELTS score 9 band in IELTS and GRE score 332 out of 340. So like this. Ikramullah, yes, yes, no need to say please. My question, please reply me, sir. Don't call me sir, bro. What are the key genomic alterations in TNBC? How can computational tools help identify them? How can machine learning models be used to predict treatment response in TNBC patient? What computational method are most effective for drug repurposing in TNBC? How can system biology approach uncover new therapeutic target for TNBC? Why, wow, Kram. Question is fantastic, but I'll answer all these questions one by one. Listen, first question is what are the key genomic alterations in TNBC? In my PPT, I explained TP53 mutation, BRCA1. Actually, on BRCA1, I am working right now. BRCA1 and BRCA2 mutation. And PIK3CA mutation, EGFR amplification, RB1 loss, MYC amplification, AR, androgen receptors mutation. So these are the mutations. So These are the key genomic alterations in TNBC. How can computational tools help identify them? You just need to get data from public repository first of all. Then you identify biomarker using ML based tools. You just go to GitHub, put your requirements, you will get tool. So you need to change something and all, or what computational method are most effective for drug repurposing in TNBC next generation sequencing. Even Dr. Sarin, a very senior scientist of Tata Memorial Centre Tech and associated with the UK Medical Centre London. So they are working on next generation sequencing NGS. So this is one of the most preferable computational method repurposing in TNBC. So you go through NGS and machine learning based how can system biology approach uncover new therapeutic system biology approach multiomics analysis को integrate करो pathway analysis and network analysis dynamic modeling and CRISPR modeling and all like this. So I hope I answered all your questions, Ikram. Any other question, please? Yes, Nabamita.
Okay, I think there is no other query left. You have answered well.
Hmm, that's
So
very good.
still, I'm going to give you time that if you have any other query, you could post if you have anything left
If
that is
I missed
undiscussed.
any question, I'm really sorry. You can put it again here. I am very willing to answer those.
I think no other question is left
Yes,
from the side of participant. So I'm going to
yes,
conclude
yes.
this session. So, dear participants, we extend our heartfelt thanks to Mr. Nishan for his invaluable insight during today's webinar. And we also thank all the participants for their active engagement contributing to the success of this event. And thank you, everyone. We appreciate you being here. And the participant, I have one announcement for you. Please make sure to fill out the feedback form so that we can improve our future webinar. I have uploaded the feedback form into the chat box. Kindly fill that feedback form that we could proceed for further improvement and improvisations. Thank you so much, sir. Thank you so
Thank you.
much, participants.
Bye, everyone. Bye.